Hello. Can you folks hear me in the back? Thank you so much for coming. The folks that are just walking in after you grab your lunch, we have plenty of space up here. Really thrilled to see the turnout here. In the interest of just keeping things moving, I'll kick things off because there's a lot of really interesting discussion coming up. I have 3 reasons I'm really excited about today. First, it's so great to see so many old friends and new friends here to learn about the myome product and some of the research we're doing. Second, I'm doubly excited that today is the first day of our myome rare disease product launch, and we're thrilled to be able to share that with you. I know the team has put a lot of time and energy into this, and we're excited to show you what we have. And third, it's an opportunity to celebrate some really outstanding science and outstanding scientists who will be sharing more about how we're looking to transform rare disease diagnosis in the future. And I couldn't think of 2 better people to share that than Danny and Stephen. So I'll introduce our other speakers after I give a short background on myome and introduce the rare disease test. First, to tell you a little bit more about myome. Or actually, before I get there, so we have lunch, and there's still a few more seats up here. Logistics-wise, we're probably not going to have time for questions afterwards. Please see Danny and Stephen up here at the end of the session. And the myome team, if you could raise your hands for the group, the myome team. There we go. You're seeing them in the back. If there are questions about myome's product, and some of the questions that come up from our discussion, and we don't have time to go over it today, please see us at the back. Also, we have our booth, which is, I believe, 507. 605, which will be at tomorrow and the day after. So please do see us. We're excited to be able to share more about what we have to offer, but we also want to make sure we have time to tell you what's coming next. So let me start by telling you a little bit more about myome and our product. I co-founded myome along with a number of very talented people about a few years ago because of some problems I was seeing as a clinical geneticist. These are problems that are not new to this group. Testing is narrow. It's hard to go from one test to the other. There were multiple barriers in place for understanding that information, and it just wasn't simple to interact with. And at myome, we've been spending the last several years really trying to imagine how we can make this information more accessible, more portable, so it's available and actionable over someone's lifetime. And this has really permeated our mission, our culture, and the whole way we approach things. Over the past several years, we've kind of reimagined how we interact with genetic data from the ground up. It starts with our platform. We start with whole genome sequencing on a PCR-free backbone to make it easier to reflex from one product to the other. We've also had to build an entire infrastructure for how we can make it possible to pull down reports from that genetic information. Those of you who have used clinical genetic testing companies know that there's some friction sometimes when you're trying to go from one test to the other. That doesn't make sense. And there's a lot of work that goes on behind the scenes to be able to build that infrastructure up. This results in 2 things. One, the ability to go from one test to the other becomes much cheaper because you have that data available as a lookup. And 2, it becomes much faster. I'll share a personal anecdote. You know, I have a genome. My genome's on the myome platform. And I was able to pull down my pharmacogenetics results in less than 5 hours. That's hours, not days or weeks. And it just shows how we can start to interact with genetic data differently if we have this information and the infrastructure around it to be able to support it. The third area that we've really had to think about how we go from the ground up is how we interact with our customers, our patients, our physicians, and what support we provide them along the way. And you'll hear some of this come up in some of the way we've approached our partnerships, both with physicians as well as in terms of genetic counseling that we provide to patients when they need it most in situations where access is an issue. So that's myome in a nutshell. Again, we're really focused on trying to build this platform that can make it easier to interact with their genetic information and pull that information over time. Over the past few years, we've had the chance to assemble a fantastic team. You can see some of our investors on the right-hand side. We've got a fantastic team. Some of our co-founders include, for example, Matt Rabinowitz, who's the chairman and co-founder at Natera, who I believe is speaking in the room next door. So there's an experienced team who knows how to execute and build genetic tests. We've also had the chance to be able to interact with very large partnerships. And I'll just highlight two examples here. One is in the state of Alabama, where they've had a really bold initiative to be able to provide genetic testing and that resulting information to the broadest population possible, the entire state of Alabama. They're sequencing tens of thousands of people in a project that's spearheaded by Southern Research. We're excited to be able to partner with them as their genetic testing provider, and it's really helped us think about how we think about access and providing information and logistics across an entire statewide initiative. On the flip side of that, we've had efforts on technology, leveraging work that we've published in Nature Medicine, JCO Precision Oncology, and a number of other areas. There's really focused on how we provide informed risk information to those who may have family history of a genetic condition or a genetic syndrome that may predispose to cancers, but may not have a positive pathogenic variant. And that's something that we've commercialized with Natera as part of a polygenic risk score that we've launched with them. So just giving a flavor of the kinds of ways we've been seeing our vision of genetic information being reanalyzed and reaccessed over time take flight. There's also a number of publications and clinical partnerships that I'm happy to talk about afterwards, but needless to say, it's been a really exciting time at Myome. Let's double click on what we're doing, what we're here about, what we're here for, and what we're very excited about to launch today. Our rare disease test is a whole genome backbone test that makes use of many of the same capabilities we were talking about earlier. The ability to have one platform, the ability to make it possible to reanalyze or reflex from one product to another, as well as some of the logistics and other support systems that we've built as part of the work we've shared earlier. You're seeing a few of these aspects here, and I want to spend the next few minutes telling you a little bit more about the product and how it might fit in your clinical workflow or in your institution's workflow. Myome's, at its very core, our rare disease product is 3 tests. The first is called a copy number analysis test. You can think of this comparable to a chromosomal microarray. It tests for microdeletions and duplications greater than 50 kb or larger. But the distinctive feature is 2 things. One is, while it's priced similarly to a chromosomal microarray, it's run on a whole genome backbone, which provides it superior resolution to be able to detect copy number abnormalities. We've done studies of over 100 different pathogenic variants, for example, and seen 100% concordance across those cases. And we and others have done this as well. But the unique factor here is that because you have this information on the genome backbone, it then becomes possible for providers who may not be comfortable with the whole genome information to at least get people onto a platform that can make it easier for them to reflex to an exome or a genome afterwards. And so while the technical details of this are fairly similar to a chromosomal microarray, the difference is the ability to analyze genome-wide CNVs. And happy to share more information we have in our research about how we validated this approach. The second test is an exome test. Similar to other exomes that look for single nucleotide variants and indels within coding regions. Different from the exome, because we have the PCR-free genome backbone, we avoid some of the bias that comes in the process of doing an exome capture, which can affect copy number variant calling. And so that's another test that's available, again, on the genome backbone for those folks that are interested. And the third test that we're offering is a genome test. And this is a genome analysis that covers the coding variation as well as the non-coding variants, some of which you'll see a little later on in the talk. Things that are on the very near horizon for us include the repeat expansions like Fragile X, and talk to one of us if you're interested in learning more about that. That'll be rolling out a little later this year. And this is just one example of the kinds of iterations and the kinds of capabilities we're looking to add to our genome platform. So 3 products. They're pretty similar to, I think, what many of us have seen. Let me just highlight a few examples that come through in the process of using this approach. One of the things that we've encountered with, in speaking with different providers and different practices, is that there are a number of geneticists and genetic counselors who will see patients who already have a chromosomal microarray. And the issue is once the individual has a chromosomal microarray, that testing information, all that data is lost. because it's not useful in generating the exome or genome information that we'd have afterwards. Using our platform, which starts with a genome, reports out chromosomal microarray information, patients can come to the geneticist with that genetic information on file, which means we can provide that information much more quickly and much more cheaply. To give you a sense of the order of magnitude here, the self-pay price for an exome or a genome following someone already being on the myome platform is around $250, which is a very low price. And we can do so because those individuals are already, as part of the myome platform, they already have the genome information available. And so we've had multiple institutions come to us and say, you know, our developmental pediatricians and pediatricians don't feel comfortable with ordering the genome. This can be an opportunity for them to do so while still having access to that genetic information afterwards. Please see our team for more information on pricing for duo trios, and we'd be happy to talk more about that and how it might work with your institution. The other advantages, which I think this team is probably very well aware of, is the ability to, on an ongoing basis, re-query and reanalyze the genome. And this can be relevant not only for new symptoms that may arise, but also new information that comes in the literature. And I won't go into it in much more detail right now, but just know that the same infrastructure we've built to make it possible to pull down more information on the genome really feeds into this process as well, which means we can do it more cheaply, more effectively, and more comprehensively as part of our roadmap. A few more details on the process of ordering tests, and again, please see our team if you're interested in learning more. We work with commercial insurance, Medicaid, Medicare. For state Medicaid programs, we don't balance bill. Have a conversation with us about the managed Medicare. We have self-pay options as well, like I shared earlier, as well as some financial assistance programs for those people who may be interested in testing but may not be able to pay the charge. The workflow that we work with is very similar to, I think, how you may be used to, with a few unique advantages. On the provider side, we have a portal, which is very seamless to go over. We're happy to walk through it at the booth. Logistics-wise, we ship a kit to the patient. That information is, the conversations on billing can be had with the patient to try to streamline the process and improve access. And reports come back in our portal. There's a few other details here that some groups have been very interested in, so I'll make that available. You know, some groups have struggled with certain areas to provide post-test counseling, and so we make that available as standard as part of our testing. In addition to that, some institutions are very interested in building their own biobanks, and so we provide back, in certain cases, that genetic information so that these institutions can help build that information. Okay, so that's the product today. It's a genome backbone, 3 products, which make it easier to access and receive genetic testing that we believe is standard of care. The next few slides I want to start introducing moving beyond the standard of care. And this is where I'm really excited to have Stephen and Danny share more about the vision of how Myome is thinking about this. Myome's research team has been very active in thinking about the next generation of testing. We have a number of different research projects that, instead of going into much detail here, I'll let Danny and Stephen speak to. I'll just start by saying that we have a strong interest in long-read sequencing because of the comprehensiveness it provides, the additional detection of structural variants, as well as methylation, which we'll hear about a little later. We have ongoing research efforts that can capture this type of information. You'll see plots like this in the next couple talks. In addition to that, we have a whole separate program on advanced interpretation that I'd be happy to go into. I just don't have time right now. But the 30-second version is that this is an approach that can improve the diagnostic yield and make it easier for individuals with a rare disease diagnosis to get diagnosed on demand, as opposed to having to go through a number of hoops to do so. I want to conclude the research section with just 2 studies that I want to make available to this group. The first is, because we see all of this research translating into a clinical platform, we want to make this testing available to the patients who use the Myome test. And so the first research program is available to those who are undiagnosed following a Myome clinical genome. As part of that process, we have you can reach out to clinical@myome. We can talk more to you about what potential options are available. But one of those options include a research study where we're looking for non-diagnostic cases on the Myome platform that will then run on multiple different analyte approaches, including long-read sequencing in-house, to be able to provide a result. And all of this is with the eye towards bringing this long-read sequencing into routine commercial use. So see us at the booth or talk to us if you're interested in this program. The second program is more focused, and this is really focused on methylation signatures. We see this as a really important area for rare disease diagnosis, especially in individuals with intellectual disabilities. We've developed several algorithms to work in this area, and we're working on validating that in a clinical trial. If you are working with individuals with, for example, Kabuki syndrome, CHARGE syndrome, SOTOS, or others, please see Lindsay or myself. Lindsay, can you raise your hand? Yep. Or myself, and we'll be happy to talk more about this study. You can see some of the details here. It involves collecting some blood and sharing some genetic testing results, but this is all to be able to provide this type of test back to the community as part of the Myome platform. So I hope we've had a chance to talk about kind of where we've been, which is narrow, traditional testing, which involves a lot of barriers. Where we are today, which we're very excited to launch, our Myome rare disease offering, which helps to break down some of those barriers by running genomes on a genome backbone, by providing information that meets patients and providers where they are, and makes it easier for people to get what we believe is standard of care testing. Where we're headed, I think, is the subject of the next two talks. And I think this is really focused on advancing the diagnostic yield using different technologies that I'll let Stephen and Danny talk to. We'd love to meet with you. We'd love to have you order our test. We won't get things right necessarily the first time, but we're going to learn very quickly with you, and we'd love to find some key partners that are willing to join us on this journey. So thanks so much for your time. That concludes my talk, and I'll hand it over to Stephen. While Stephen's walking up here, I'll just say it's my pleasure to introduce Dr. Stephen Montgomery. I'll let you get set up here. Stephen is a professor of genetics, of data science, and pathology at Stanford. He leads a number of research initiatives, including the Gregor program, where he's the site director at Stanford. But I think, he's a huge advocate for rare disease community. But one anecdote I'll share is that in addition to his brains and energy, I think it's pretty unanimous across the Stanford community, and I think most people that work with Stephen, that he is an exceptional collaborator. He's very generous with his time and ideas, and he is just a very kind individual. And we've been very fortunate to have him on our scientific advisory board, and very excited to hear what he has to say next.
Gosh, that was very kind of you. Thank you. I can use this right here. That sounds all right? Okay, I can take the mic. Okay, perfect. There are some seats up at the front if people want to grab them. Can you hear me now? Test? Yep. All right, thank you. So I'm going to talk a little bit about some of the research that we're doing as part of the Gregor Consortium, which is a consortium focused on developing novel strategies to solve rare diseases, and spend a little bit of time speaking in more detail about what's happening at our Stanford site as part of Gregor. So I think all of us want to solve this problem and think about how we can increase the diagnosis of rare diseases. And we know currently, roughly an estimate, about 50% of people that get clinical genetic testing don't receive an answer as to what is the genetic etiology of their disease. And so really we want to get that number up to 100%. We want to think about what are the strategies, what are the novel tests and assays that we can do that will get us there. We also, as part of this, want to think about how we can reduce the time to diagnosis. And so this is a reference saying the average time to diagnosis is about close to 5 years. As we can accelerate different technologies and use them in different settings, we should be able to get that number down. I'm just going to ask, I can hear some feedback here. Is it okay for everyone out there? Yeah, great. Okay. So after the exome, we know there's a lot of different strategies. We actually know that's true after the genome, too, as Akash just pointed out. Danny was part of this fantastic review that said, there are multiple ways to sort of navigate through, the maze of different assays that we can do afterwards. And so, you can think about multi-omics, you can think about long-read genomes, OGM, you can do reanalysis after a period of time. But, a lot of those different assays and approaches are sort of context dependent. What do you have available at your site? What can you afford to do? What research protocols can you put participants on? And I think what we need as a community is we need a way to look at this maze of all these different strategies and figure out what is the specific optimum strategy that's going to be effective for any patient that we see. And so foundationally, this is what the Gregor Consortium was established to try and address. This is a range of different sites across the US who have gone together to think about those unsolved rare disease cases where clinical genetic testing didn't provide any answers. And if we could throw different assays at these individuals, what would we learn? And how could we scale those assays and develop standards so they get used more regularly in clinical practice? Our site at Stanford is mostly focused on the Northern California clinical genetics labs and the medical genetics laboratories. We've had partnerships with UCSF, Davis, Kaiser, et cetera. We reach out through our medical genetics alumni network, but we are very interested in working with anyone here who might have referrals of particularly hard cases. Recently, we've established remote sample collections, so we realize there's a challenge for many people with rare diseases to make it to an academic medical center. And so we want to be able to send mobile phlebotomists to where they live, to be able to collect samples to do a range of different assays. And there are other cohort studies that we have in place. We also have really exciting industry partnerships. So my own being one partnership where we're exploring new assays for looking at long-read genomes for some of our participants. Our recruitment statistics so far over the last couple of years are we've enrolled 265 probands. So this is balanced, mostly 50-50 female-male. Most of these cases are pediatric. We have some retrospective cases as part of the Undiagnosed Disease Network, but most of these 80% are prospective cases, so new people that are coming into our site. And as I mentioned, what we're doing is we're throwing everything that we can think of at these individuals and their family members to see what we learn. So our core assays are short-read whole genome sequencing. We're doing long-read whole genome sequencing and adding methylation from that. We're doing bulk RNA sequencing from blood, metabolomics, attack sequencing, which is an assay of chromatin accessibility to see, are there interesting regulatory effects there. But we've also been adding proteomics through Olink and developing some custom RNA-seq solutions that allow us to look for rare transcripts in the blood that might, canonically not thought to be expressed. And one example of that is like the DMD gene, where people typically get a muscle biopsy, but it is lowly expressed in blood. And so if we can enrich for that, we might be able to do things that are less invasive for our research participants. So I'm going to talk mostly about the long-read whole genome sequencing and methylation and touch a little bit on multi-omics as well here. Gregor, where it sits with respect to the UDN, we've been sequencing long-read genomes, but we actually have access to many long-read genomes over a range of different collaborations. This is just showing those. In total, it's close to 1,600 or 1,700, long-read genomes. One of these is a collaboration with Danny, where there's Oxford nanopore sequencing of the 1,000 genomes population. So it's really having these large long-read whole genome sequencing cohorts is allowing us to define what is actually a rare variant in the cohort. And we've developed experiences as well with confirming long-read whole genome sequencing findings. I'm not going to walk through each one of these cases here in the examples, but most of the cases that we've seen so far help us confirm the phasing of biallelic effects. So we can, in many cases, the clinical short-read genome sequencing, it could identify the variants, but we don't get the phase to understand what's happening there. So there's 3 of those so far that we've had contributions to, and we've had reports from clinical labs that established the variants being in trans. So I'll talk a little bit about one sub-cohort of this, which we studied a bit more in detail, where we're combining long-read whole genome sequencing with multi-omics. We had 70 exome-negative patients recruited from the UDN. This was 57 affected individuals and some of their unaffected relatives. These were a range of different symptoms that these individuals had, so we weren't looking at any specific rare disease here. They had Illumina short-read whole genome sequencing and some with whole exome sequencing, but in every case, there is inconclusive evidence to provide a diagnosis. So we ran nanopore genomes using the Oxford nanopore Promethean R9 flow cells. We got about 15x coverage, 19 kilobases of read length, that's our N50. And the goal was to find, are there novel structural variants that could be explaining some of these cases. As expected, and maybe many people have seen these types of plots before, when you do long-read whole genome sequencing, you detect more insertion deletions and structural variants than short reads. So this is just showing a comparison plot between long-read data and short-read data, what the distribution of discovered deletions and their sizes are, and insertions as well and their sizes. And you can see definitely for insertions, you're finding some longer events. We could merge this with our original 1,700 individuals where we had, where we collected long-read genomes to start thinking about a larger population reference that gave us allele frequencies. So, you know, there isn't really a nomad for long-read whole genome sequencing yet. So this is giving us those first insights into what are these interesting rare events in our patient population. And this is just looking at the SV allele frequency for what we found for the long-read data. And it sort of maps what you'd expect in terms of a human population allele frequency spectrum. We're seeing, you know, many more rare events for all categories of SVs found with long-read whole genome sequencing. And we can compare the number of these discoveries as well between the different technologies, and so we're finding more deletions, more insertions. It's a little bit different for duplications and inversions as well, but you know, in many cases, it's about an order of magnitude more events that we're discovering. So one thing that was also special about this cohort is we had RNA sequencing that was available for each of these participants too. So not only could we say, here's a rare structural variant, we could ask, is there any evidence from the RNA sequencing data that there's an effect on gene function nearby? And we think this is actually a pretty reasonable strategy for prioritizing interesting SVs. So if you see that functional effect, you're more likely to want to consider it downstream instead of filtering it out. And obviously, it does depend on the tissue that you're looking at. So RNA sequencing, there is a tissue specificity to, you know, where genes are expressed. And so it's possible to miss things. But I'm going to show you in a few cases where this actually worked, combining these data. So we had this for muscle, fibroblasts, and blood for a range of different patients. We had outliers that were collected, so really aberrant expression for each one of these individuals in the cohort. And we could see that these rare structural variants found through long-read whole genome sequencing were enriched near expression outlier genes as a function of the strength of that outlier effect. So what this plot is showing is as you go along the z-score threshold, as you get the stronger outliers, you're more likely to find there's a rare inversion or insertion or deletion or duplication near that gene. So there's some evidence that the rare variants are driving these extreme effects. We could also see that the direction of that effect on expression made sense in many cases. So if you just actually look at the first two here, which are deletions and insertions, it's no surprise that a deletion is enriched for under expression events. So if you're getting rid of the gene, obviously it's not expressed, right? And for insertions, where you might be duplicating part of it, and actually duplications beside it, you see that enrichment for overexpression outliers. So those extreme events that are going up might be caused by some of these events. So we thought a little bit about, okay, we've got these observations from the RNA-seq data and these long-read whole genome sequencing SVs that we're getting out. Is there a way to actually more effectively prioritize this data? And so we've been working on developing models that simultaneously take personal omics data and genome annotations for structural variants to come up with prioritized structural variant gene pairs and candidate diagnoses for our participants. And this model is called Watershed SV. We've trained it against genome-only models, and we've actually seen, you can see these blue lines here, increased performance for these models in comparison to the red lines, which are your genome-only model. So the more interesting thing there is that we can use it as an additional filter to prioritize SVs in rare disease cases. And this is just one example this is looking at all the data in aggregate across our cohort as we consider rare SVs close to the genes, those with high CAD-SV scores, those with HPO term matches, expression outliers, and the combination of that data, we increasingly reduce the number of candidate SVs that we have to look at, a number of genes as well. And we also get a smaller subset of patients where this information is giving us some interesting candidates. The other interesting thing to note, and the value of going forward with long-read whole genomes in the future, why I'm excited about it, is actually a lot of these prioritized variants are unique to long reads. So things that we wouldn't have seen if we run a short-read whole genome. So this purple bar showing the long-read unique set of structural variants that were also prioritized by Watershed SV. And you can see this is, we're finding somewhere on the order of 600 of these events compared to that CAD SV model, which is your genome-only model. Okay, so I'll give just an example of how this all worked out. So we were able to run Watershed SV on one particular patient. These were sisters that were age 68 and 69. We had one of them as a proband. We could winnow down through the different SV gene pairs that were prioritized and use the Watershed SV model with a posteriori of 0.6 to get roughly on the order of 6 to 7 interesting SVs. One of those happened to be in this gene FAM193B, which was interesting for us to follow up. It is there's a tandem repeat expansion in the 5' UTR of this gene. And so these plots are just showing the position of the variant with respect to the UTR that's there. We also were able to look at the repeat copy number. And so you can see the proband and the affected sibling, as well as an unaffected parent, have a considerable number of repeats compared to the population at large. So this became an interesting candidate for us, but also because we have this expression, we could go back in and see what was Watershed prioritizing. And actually, both the affected sibling and the proband were overexpression outliers in the blood. So it's a really strong effect there. They have a z-score of 10 compared to the rest of the population. And this type of model, like using Watershed, gave us better posteriori information on prioritizing this variant than just having the whole genome data alone. So that's neat when you have RNA-seq. In many cases, people don't have RNA-seq along with their samples. And so we've also been investigating how can we leverage methylation, and Akash was pointing to this as well, which we get from long-read whole genome sequencing into these models to help diagnose patients. And so a neat thing about both Oxford Nanopore and PacBio is that you can measure methylation status off of them. And we know there are definitely episignatures which are informative of different rare diseases, but also localized methylation patterns that might inform an interesting effect. So, some of these are printing disorders or epivariants, but this is sort of what the data looks like. So when you get it out from long-read whole genome sequencing data, you can get these methylation coverage plots. In this case, there are some samples where you see information on both haplotypes about methylation status. So we can actually create those backgrounds of methylation across different CPGs, across these long-read genomes, and stratify methylated versus unmethylated loci. But we can begin to actually start to compare our patient samples to the population at large in the same way that we did with RNA-seq. So here we could start looking at patterns where there might be a segment of CPGs which look really aberrantly methylated compared to the population average. And so we've been developing methods to try and segment out these methylation patterns and identify where they might occur in our research participant samples. Basically, trying to find contiguously aberrant regions across these loci, and then using them to come up with z-scores, again, in the same way with RNA-seq. So saying this is something that's undermethylated or hypomethylated or hypermethylated in those contexts. And so this has been useful as well in the context of work at, collaborative work, where there was a couple of sisters who were suffering from a rare disease, a range of different phenotypes. Some of the interesting things that there were 7 standard deviations away in terms of overgrowth and early onset obesity. And so they underwent this long-read multi-omics workup where they had a genome from the nanopore sequencer. They also had methylation called from that genome. But we did have RNA-seq as well to look at some things after. And the first thing we found using this type of methylation outlier approach is that there is a really interesting methylation outlier identified in the 5' TSS of the GNAS gene. And so some of you may have heard this story as well. This is an imprinting disorder gene. And the methylation outlier marked the loss of imprinting for that GNAS gene. So this is what you'd expect in the control. So you see, across this sort of middle region here, it's 50-50 methylated versus unmethylated. Whereas in our patient samples, you can see that they're completely unmethylated. So denoted by the completely blue segment that's in there, right? And so there was also a rare deletion that was found nearby this methylation outlier that removed a cis-imprinting control element. So this wasn't, at the site of methylation, but it was in the intron of this STX16 gene. And so there is a well-known mechanism where there's a long-range effective deletion that leads to loss of methylation in the GNS gene at a distance of 300 kilobases away. So this SV is taking out this imprinting control element and causing a change in the methylation status of that gene. So we can look at the RNA-seq, and we actually saw that there's overexpression, which confirms the expectation of a loss of methylation. Some methylated regions are typically silencing genes. So that was a nice confirmation of this effect using this data. And actually, we get all of that from just the long-read whole genome sequencing alone. So we've been trying to put these methods out there. This is just an example from a talk we gave at ASHG recently of the methylation outlier approach. We've now put this on GitHub so people can use this with their long-read data. So this is a new thing that's available that people can run now if they have that kind of data, and we're happy to help you as well. And so in summary, I just want to say, we are solving cases with long-read whole genome data. We are giving back, clinically confirmed variants. Most of those are through accurate phasing. We are starting to begin to use multi-omics, both RNA-seq and methylation outliers, in combination with long-read whole genomes to improve the prioritization of different rare SVs. And with that, we're identifying interesting and novel diagnoses. And so really our next steps are like, how do we scale this? How do we make this available to more patients in terms of these methods and tests? And we're definitely excited working with Myome there as they're putting some of these tests out there into the world too. So I'll leave it at that, and I'm happy to take questions at the end. Yeah. Oh yeah, okay. And there's an acknowledgement slide, sorry. Yeah.
Thank you very much, Stephen. That was a really great overview of different ways of solving the undiagnosed. And we're very fortunate to have you on our advisory board. Next up, I want to introduce Danny Miller, Dr. Danny Miller. He is an assistant professor at the University of Washington. As he's coming up here, I'll just share, it was about 2018 when I first met him, when I was a resident at Stanford in genetics, and he was coming to interview. And I didn't hear from him until 2020, when he published, there's some papers out there that make your hair stand on your arm. He had one of those papers. There was a paper in AJHG eventually that was published that was like a tour de force on long-read sequencing, showing a whole number of different technology use cases in one paper. It was really a fantastic paper. I encourage you to read it if you haven't. But as a clinical physician scientist, he's very bold. You'll find him in Kenya or Europe, working on sequencing, rare or undiagnosed populations. He also has as a technologist, a knack for making pretty bold predictions. And I'll say after about 7 years now of knowing Danny, he's not one to bet against.
Thank you, Akash. I think I'm going to use the microphone because if there was feedback, I'm an assistant professor. I appreciate feedback still, but when you're speaking, it's a little hard. So first off, thank you for the opportunity to speak. Thank you all for coming to the session. It's also really flattering to speak after Stephen. He's somebody that I've looked up to for many years, so I appreciate this opportunity. So my goal today is just to talk about how we've used long reads in various ways to look at epigenetic modifications to diagnose disease, better understand disease, get a sense of what's changing in a genome that could explain an individual's phenotype. I am going to start with slides that I actually often start with. If you've seen me give a talk, you've probably seen this. My lab, what we think about is how can we use long-read sequencing to really change the diagnostic process or diagnostic odyssey. So the first thing I'll say is that I do think long reads will dramatically change the way we do clinical genetic testing, and that's going to be now in 3 years because I started saying this 2 years ago. It makes testing more equitable. Akash did a really nice job of kind of talking about how do you lower barriers to testing for families, how do you bring people, kind of get them the answers they need in a short amount of time. And then I do think this happens if you give away other types of data. Even if you pay to generate the long read data, it is worth it, even if you give away things like short read sequencing or array-based data. So Akash had an image similar to this, and it's kind of a very basic, the linear process of the diagnostic, not the odyssey, but just the diagnostic process in the clinical lab. I think the diagnostic odyssey includes more than just the testing, right? It's the visits to the specialist, it's the additional lab tests, it's the imaging, things like that. So we go through this with families in the clinic, right? We order a test, we get a negative result back, we order another test, we try to read the PDF that was scanned 3 different times and maybe it was duplicated and we can't really understand it, until eventually we get to the point where more than half of individuals we see in clinic are undiagnosed, which is disappointing and also surprising, and likely just because we're not looking at the right things when we do this testing. So my lab thinks about how do we use new technologies, long-read sequencing, to change the process, to change the odyssey, or change the testing process, and get people to answers quicker. So just really quickly, I'm going to give a what do I think about when I think about long-read sequencing, what's the advantage? And the first thing is it really simplifies the analysis of complex regions of the genome. So if you just think about a simple repetitive region, a CGG that's repeated multiple times, if you use short-read sequencing, you may not be able to actually identify the total number of repeats in that region or the actual sequence composition of those repeats. And those can be clinically relevant. FMR1, for example, the propensity to increase in size can depend on if there are interruptions. Huntington's, for example, and then others. RFC1, knowing the sequence of that repeat is important. Long reads solve this problem or make the analysis simpler by just being longer than the length of many of the repetitive regions. You can just basically get a read that spans the whole region. You're going to know the sequence of that region and the length of that region. And then I'm not going to go through all the numbers. We do know that short reads, they used to be a lot higher accuracy than long reads, but today I would argue that long reads are actually higher accuracy in certain contexts. And then the costs have come down dramatically to where it could cost less than, or about $500 or so to get a long read whole genome at this point. The important thing here, and kind of the thing I'm going to focus on for the rest of the talk, is that these are information-rich reads, so they contain the modification information that we can use. From a structural variant perspective, it's pretty striking the advantage that you get with long-read sequencing. We know that we identify fewer than half of all the structural variants present in any one of our genomes using short-read sequencing technology. So these are regions of the genome, these are variants that are clinically relevant, they're disease-causing, and we miss them when we just use short-read sequencing. So an example of combining all these together is actually from that 2020, well, it's from the 2021 paper. The reference is on the next slide that Akash just mentioned. So this is a family that was seen in Seattle. Heather Mefford was the senior author on this study. Essentially 2 kiddos who had phenotypically Baratell-Scott syndrome, but negative clinical testing. We knew that they had a deletion over the gene associated with it, which is XYLT1, but we didn't know what the second hit was. They solved it by identifying a repeat expansion in the 5' UTR of XYLT1. This is the southern over here, and this shows the expansion in the proband of one of these 2 kiddos. It took years and a lot of testing to figure that out. So we asked a very simple question, which is, could we use long reads to replicate that entire process? And the answer was yes. So here's a cartoon of the gene, the 5' UTR. This is the expansion. And then we see that in the proband, we're able to capture reads which fully span the expansion. They're red because they're methylated. There's a gray one here because we used a methylation software that didn't always convert reads back then, but it does today. And then in the parents, you can see that mom has about 2 separate clusters of reads and dad has just 1. If you go look at the IGV plot, you can see that in the proband, there's an expansion and it's all methylated. In mom, you can see that some of her reads are methylated, whereas others aren't, which I thought was interesting because it tells you that even though she's not affected, she does have some cells that have chosen to inactivate the haplotype that has the expansion or the premutation, and then dad is normal. Point here being that you can get multiple pieces of information from a single data source. Long reads gives you the repeat, it gives you the sequence, and it gives you the methylation as well. I'm not going to go through this in detail. I just want to point out that at this point, the historical concerns about long-read sequencing, I think, have been largely resolved. So it includes things like the cost, the error rate, the complexities with data analysis, and then, the sample source. You don't need micrograms of DNA to do the sequencing. You can do it with very low amounts of input. So what are some ways that we've used long-read sequencing to solve some challenging cases? Before I get to that, I just want to talk about how we've been thinking about using it in a control cohort. So I'm going to show some data from the 1,000 Genomes Project. As Stephen mentioned, this is an effort where we're sequencing right now 500 or so. Or I'm sorry, we're planning on 800. We've sequenced 500 individuals from the 1,000 Genomes Project on the Nanopore platform. We put that data up on AWS. You could go download it today if you wanted to. It's the genome alignments, the assemblies, and then also the raw data. Careful, it's about 500 terabytes of data. But it's there for people to use, and we also have summarized data up as well. So we've gone through and looked for patterns of methylation in that data to try to understand what are we seeing. So here's just one example. If you want to look at X chromosome inactivation, you can plot CPG islands along the X chromosome. This is not every CPG island. There's select ones that we know are differentially methylated. But we see that in one cell line, for example, we get a nice mixed pattern of inactivation, which is what we would expect in a XX cell line. We can look at the IGV data, and we can look over a CPG island in FMR1, and we can see a nice mixed pattern here with haplotype 1 and haplotype 2. And this phasing is actually done using the SNPs from the individual. We didn't need parental samples to do this. We can then look and ask how many of the samples have skewed X inactivation. So here you see that there's a group of reads a group of dots which are basically near 0, and a group that are somewhere around 75%. This is what we see when we see skewed X inactivation. There's some technical reasons why it never goes completely to 0 and why it never goes to 100. So this is kind of the range that we would expect for someone who has completely skewed X inactivation. And then you can go look at the IGV view again, and you can see that for this haplotype is active, it's blue, and then this haplotype is inactive, it's red. We can use that methylation data then to do some interesting quality control of the samples that are sequenced. So this is just a PCA plot. It's not pretty, I apologize. But the point here is that we have long-read sequencing data from individuals from both blood and cell cultures, and then we have the reported sex. And we can see that they cluster as we would expect, where blood clusters together, cell lines and cell lines. But if you squint, there's one outlier. There's a triangle here. So it says that it was reported to be a male, but it's clustering with these female samples. So this is just looking at individual CPGs. If we look at the CPG islands, we get cleaner data, but we still have this one outlier which sits in a group that we didn't expect. This outlier. And then finally, if we just do this plot as a whole, what we find is that this is actually a cell line that is now 45X. So it started out as a 46XX cell line, and it's lost one of its X chromosomes. We did confirm by looking at the short read data that this individual did lose an X chromosome. It's probably an artifact from the cell line. It's not necessarily a feature of the person who contributed this sample. So we can use this to both look for skewed X inactivation and also do quality control on the samples that are sequenced. So why do we do this? Well, one of our long-term goals, and I'll come back to this at the end, is to build long-read sequencing-derived methylation classifiers. This is just an example of what a heat map might look like, where you have individuals who have a particular condition, and you group them by methylation pattern to determine whether or not they have that condition or to resolve a VUS. And we think the advantage here is that most of these have been done using array-based approaches, which are about 800,000 CPGs. With long reads, you get to look at all 44 million CPGs. So we think that not only will the classifiers work, or we know that they work, we think we'll be able to get better resolution of individual cases, of individual conditions using more data. And this is something that's obviously actively under development. So how do we use methylation to either identify a condition or to better understand a condition? This is a story that came from Heather Mefford. She's now at St. Jude. They had an individual who, shortly after birth, developed epilepsy, went through the kind of standard genetic testing process, had a genome done, and no pathogenic variants were found in that individual. Unfortunately, the individual passed away and was in one of Heather's research studies where she did methylation arrays on all of the individuals who had epilepsy. And when she looked at the data, she found that there was one outlier, which is the red line here. So this is methylation fraction along different positions of chromosome 13. And you can see that this one sample always sits a little bit higher than the others. Heather asked us to take a look at it using long reads, which we did. So this is just a random segment of chromosome 13. And we see over the CPG island, you get some reads that are methylated and other reads that are not. Most reads are not methylated. That's kind of an interesting finding, but of course one nice feature of long reads is that you can phase the reads. And when we do that, what we find is that on one haplotype, all the reads are unmethylated, but on the other haplotypes, some of the reads are methylated, whereas others aren't. And that's actually an important clue in this case. So one reason that you might get methylation along a stretch of a chromosome, only on one chromosome, could be something like a translocation, which is actually what ended up happening in this instance. This is an X13 translocation. It was missed by whole genome sequencing. It was missed by reanalysis of the research data as well. And we identified it both by methylation and then also by looking at the structural variant calls from that individual. So you can see just right here on the X chromosome, the reads don't go all the way across. This is the translocation breakpoint. The other haplotype looks just fine. And then typically when you have an X autosome translocation, the structurally abnormal chromosome is the one that's active because you don't want to inactivate your autosome. But in this particular case, the opposite happened. And sorry, I didn't show you I don't have the methylation view, but you just have to trust me on that. Okay, I'm going to give you one example of how we've used methylation to better understand how an individual was impacted by a particular condition. I'm going to talk about a disease called incontinenta pigmenti, or IP. This is an X-linked condition. It's caused by variants in a gene called IKBKG. This is a very tricky gene because most of it lives within a very large segmental duplication on the X chromosome. So it's hard to sequence and analyze that particular gene. This is a screenshot from the table from Gene Reviews, which shows the diagnostic rate. So there is a well-known 11,000 base pair deletion, which about 65% of individuals carry. Sequencing will get you about 10%, and then doing del-dup will get you about 4%. And again, this is the gene sits here on the X. Most of it lives within this big segmental duplication, which is also right here. These are almost identical, which makes them really hard to analyze. This is a case that Monica Wojcik from the Broad referred to us. This is a little girl who unfortunately passed away at 13 days of life. She had kind of the characteristic skin findings of incontinenta pigmenti. She had clinical testing, which was negative. So for the proband, they did targeted sequencing of IKBKG, and then they did long-range PCR looking for the common deletion, and it was negative. What was interesting about this individual is that her family, there was about 4 generations who all had clinical diagnoses of incontinenta pigmenti but never had positive clinical testing. So the question Monica had, the question the family has, was what caused this particular condition? And I just want to point out that there's very few males on that side of the family, 1 or 2, but for the most part they're having pregnancy losses throughout. So we did whole genome long-read sequencing, and we identified, this is of the proband, and we identified an atypical deletion, which removed exons 8 through 10 in the proband. And what was nice is that we could kind of look across here and see that the rest of the gene was intact. This phased to a single haplotype. The question, of course, was why was she more affected than her other family members? Well, we first had to actually confirm that the deletion was passed on through the family, and we did have data from the grandmother, an aunt, and mom, and they all carried the same deletion. And when we went and looked at methylation in the proband, we can see that over a CPG right here, we see a mixed pattern of X inactivation. So we would have expected that she would have completely inactivated the chromosome with the deletion, but she actually has this mixed pattern where she didn't select against this particular chromosome, which likely explains the severity of her phenotype. If you go back through the family, you can see that mom does have skewed X inactivation. She's inactivated the chromosome with the deletion. Aunt, same thing, and grandma, same thing. We can look along the entirety of the X chromosome in all 4 individuals, and we can see that the pattern holds throughout the entire chromosome. So the proband has this mixed pattern, whereas the other 3 relatives have skewed X inactivation. So it's this kind of information. I mean, you can't get this with an array, right? You've got to do a specific test just to get, it's a single test to get skewing at the antigen receptor. You wouldn't be able to see the entire genome. So this is, I think alone an argument for using long reads in situations like IP. Okay, really quickly, we've at at University of Washington and Seattle Children's started thinking about how do we start implementing long-read sequencing clinically. So one way that we're thinking about doing it is as a test for imprinting disorders. This is a collaboration with our clinical lab at Seattle Children's with Kate Paschal, who's the director of the lab. As many of you know, that testing for imprinting disorders is complex and often stepwise, so it takes multiple tests to get through the kind of diagnostic process. And this is for both Prader-Willi and Angelman syndrome and other conditions like Beckwith-Wiedemann syndrome. So we asked, could we use long reads as a single test to replace the assay? And the answer is we think we can, or the answer is yes, honestly. The challenge we had is how do you actually make the analysis of the data easy? Because it's actually very hard to go looking through IGV, trying to find variants, things like that. So what we did is we designed a study. We identified 20 individuals with either Angelman or Prader-Willi syndrome, and then 3 negative controls. And we did whole genome long-read sequencing, and then we made a single report that 3 individuals could look at and basically answer the question, is this Angelman, is this Prader-Willi, or neither? So the first thing on the report is that you can look at the copy number variation in the 15q region, and you can see here's the common breakpoints, and it's a little washed out, I'm sorry, there's some other boxes here, but you get the idea of where the breakpoints are. And you can see that for this individual, you get loss of this region of the chromosome, so there's a deletion. You can look at polymorphisms, and you can see that there are no heterozygous SNPs within the deleted region, which is what you would expect if you have a deletion. And then finally, you can look at methylation. There are multiple CPG islands across this region. The key one is this 7735 CPG, which is in the SNRF-SNRPN gene. And we can see that for this individual, the population is about 50% methylation, whereas this individual has basically no methylation. So they have not inherited the maternal region of that, so they have Angelman syndrome. I will say I don't know which one of these other CPG islands will remain. They're not all useful, so it may just be that we focus on this one with the final clinical test. And then finally, I just want to point out that we can use long reads to do long-range phasing. So 10% of cases of Angelman syndrome are caused by pathogenic variants in UBE3A. And here's an example of that, where this individual has a 3 base pair deletion, which we know is pathogenic. We don't know if it was on her maternal or paternal chromosome. We assume it's maternal just based on the phenotype. But using long reads, we could phase over this entire region. It's about 500,000 base pairs. And we could see that the deletion is on the same haplotype where there's inactivation. So this is the maternal chromosome. So how do we actually start using this in the clinical environment? What problems do we still need to solve? We've made a ton of progress, but we still do have a little ways to go. So one thing we need to solve is building better databases of variation for filtering and prioritization. Stephen talked about this a little bit. We've been doing that using the 1,000 genomes data, collaborating with other groups throughout the world, and I do think that we will see something like a NOMAD for structural variation start to come online in the next few years and eventually get incorporated into NOMAD. We also need data for methylation as well. We've started doing this, and then I'll talk really briefly about the myome effort as well. We, of course, need better automation for the clinical lab. Making good libraries for either Nanopore or PacBio is not a trivial task. So getting that automated, where it's basically blood in, library out, is where we need to get to. We need good tertiary analysis pipelines that weren't developed to analyze exome data initially, because they need to be able to take advantage of all aspects of long-read data, the phasing, the methylation, the structural variation, et cetera. We, of course, need to talk to the payers about changing their mindset about using long reads as a single test versus doing it in a stepwise fashion, and I think we're making good progress on that. And then, of course, we need good resources for counseling individuals on some of the complex things that we find using long-read sequencing. If you've never tried to talk to a family about a mosaic inversion in IKBKG that causes incontinenta pigmenti, I challenge you to do that in 30 minutes, for which you will be reimbursed for. So I just want to circle back to something Akash mentioned earlier. We're pretty excited to launch this effort to build epi-signatures for 5 conditions, Kabuki, Charge, Sotos, and Prader-Willi and Angelman syndrome. The goal here is that we can use long-read sequencing data to characterize, classify these conditions, and identify them from those genomes. So please reach out if you have cases in families who you think would be interested in participating. So with that, I will say first thank you to my lab for all the work they do, and then again, thank you for the opportunity, and thank you all for your attention.
Thanks so much, Danny. Let's hear one last round of applause for Stephen and Danny. So that concludes the session today. I really appreciate you folks stopping by. Just before we wrap up, just kind of 3 things that I wanted to highlight here. One is at Myome, we're really trying to build a platform that makes genetic testing more easily accessible and informative over someone's lifetime. We've got the rare disease test, which are 3 tests: copy number analysis, exomes, and genome. Please see us at 6:05 if you're interested, or speak with any of the folks in Myome with a Myome badge if you're interested in learning more. And the third thing is that same frontier that we're trying to push for long-read sequencing is something we'd love to partner with you on. We'd love to publish with you as you kind of use our tests and work with our research team. So please reach out to anyone on the Myome side, and they can connect you with some next steps. Thank you all for your time.